Thank you to Congreso Futuro for inviting me to come back to Chile and tell you about some of the work that we've been doing. So today I'm going to talk to you about a little bit about how we think life got started on the early Earth. Very simple forms of life, similar to this image in the background here with a cell membrane, a little bit of genetic material. And so we need to understand how structures like this could form to give rise, to allow evolution to start and to give rise to the more complex forms of life that you just heard about from Elena. So before I get into that, I want to just say that this question of how life got started is closely related to something we heard about in the first talk this session, which is there life out there? We know there are millions or billions of planets, rocky planets like Earth, that probably have water on their surface, that could support life, but we don't know. And the answer to that question depends on whether the probability of life starting is very low, if it's a very difficult process, or if it's very easy for the chemistry of the early Earth to give rise to life. If that's the case, life should be everywhere in the universe. But if it's very difficult, we may be alone in the universe. Okay. So there are two main reasons for thinking that life might be common in the universe. One, as I just said, there are many, many places in our galaxy alone that we know could support life. And also, if you look at just the history of our planet, we know that there was a long period of time between when our planet cooled down enough to have liquid water and then the first clear evidence of life, a period of about 700 million years. The problem is we don't know when in that period life emerged. Could have been almost as soon as the conditions were favorable, or if the process is very long and difficult and low probability, it might have taken hundreds of millions of years. So we don't know. There's no evidence left from that time period in Earth's history. So we're trying to do what we can in the lab to figure this out. Okay. Now, on the other hand, there are some very good reasons for thinking that it might be difficult to get to life. So, for example, anybody who studied biology and biochemistry knows that every cell, even the simplest bacterial cells in our planet, have thousands of chemical reactions going on in parallel, not interfering with each other, all controlled by very sophisticated enzyme catalysts. So how could such a complicated system emerge spontaneously? If you look at the flow of information that's behind this biochemistry, the problem is even worse, because again, in every cell on our planet, information is stored in DNA, transcribed into RNA. RNA directs the synthesis of proteins that catalyze reactions, and those proteins control the metabolism of the cell. The problem is that every part of this system depends on every other part. So, for example, you can't make DNA without help from RNA and proteins and metabolites. You can't make RNA without help from DNA and protein enzymes, and so on. So how could a cellular system, where every part depends on every other part, emerge spontaneously? That problem was so difficult that it wasn't really possible to think clearly about it until the proposal and then the discovery by Tom Cech and Sid Altman in the early 80s that RNA molecules could also act as enzymes. And so that allowed us to imagine a simpler, earlier phase in the evolution of life, where simple cells would have an RNA genome that coded for RNA enzymes that maybe controlled a simple metabolism and regulation also by RNA. So now the problem's much simpler. Still pretty complicated, much simpler. We just need to understand how to go from the chemistry of the early Earth to this simpler RNA-based form of life. Okay. So to answer a question like that, any big question in science, or actually in anything, you have to try to break it down into simpler parts that we can address separately. And so in thinking about the origin of life, we need to first understand how the building blocks, the chemicals that make up biology, could have been made on the early Earth. And then, if we have the right chemicals, how do they come together to make a living cell that can grow and divide and evolve into more complex forms of life? That second part is what my lab has mostly focused on over the last 25 years or so. And finally, we're getting to the point as a field that we can start to think about the environments on the early Earth that could have supported the synthesis of the building blocks of life and nurtured the first cells. Now, due to limitations of time, I'm going to focus on the middle question. Once you have the right chemicals, how can they get together to make a living cell? So, again, we have to break that big question down into some smaller questions, and that's illustrated on this diagram, which shows how we think of the structure of a very simple minimal cell. We need a cell membrane made of simple lipids, much simpler than the membranes in our cells. And inside, we need some genetic material. And for many reasons, we think that had to be RNA, or something very similar to RNA. So actually, assembling structures like this is very easy. The problems, the difficult and interesting problems, are how can the cell membrane grow and divide to give daughter cells when there's no biological machinery? We have to rely on chemistry and physics to do this. And the same thing applies for the replication of the genetic material inside this very primitive cell or protocell. So I'll start by talking about the cell membrane. And we know that membranes can be made from very simple molecules, fatty acids. For example, oleic acid is the fatty acid that can be extracted from olive oil. Capric acid on the bottom is a very simple molecule, can be made in several different ways in early prebiotic environments. All of these molecules, if you shake them up in water, not too acidic, not too basic, they'll spontaneously assemble into beautiful structures like you see here. So these are vesicles with a bilayer membrane. In this case, you see a big vesicle with smaller vesicles on the inside. So we've been experimenting with these fatty acid-based vesicles for about 25 years now. And I just wanted to show this one experiment where we made two populations. One set of vesicles labeled with a green dye, one set of vesicles labeled with a red dye. We mixed them together, and then this picture was taken a day later. And you can see there are still red and green colored vesicles, so they haven't mixed, they haven't fused. They've maintained their individual identity, even though they're floating around in this population. And the most interesting thing is because fatty acids come in and out of the membrane and exchange between vesicles on a very rapid timescale, these structures are made of different molecules from second to second, but they maintain their identity for weeks or months. Much like us, as we eat food, we're made of different collections of molecules from year to year, but we maintain our individual identities. Okay, so the biggest question is how could a simple structure like that grow and divide? And so I want to show you a movie that was made by Anna Wong when she was a postdoc in the lab. She's now a professor in Australia. And this is a population of these vesicles that Anna made that contain green labeled RNA on the inside and an orange dye that stains the membrane. So to make them grow, we feed them. We add more fatty acids. The membrane grows in area, and then that allows the shapes to start to fluctuate. And so if you look at this carefully, you'll start to see different shapes that are fluctuating. These vesicles are changing what they look like, and division is happening. You can see some of the big vesicles are giving rise to smaller daughter vesicles. And so the lesson from this very simple kind of experiment is that growth and division can happen just based on very simple physics and chemistry. Now, I do have to say, though, on the other hand, that there are many mysteries about this process. We certainly don't understand all the biophysical details, and so solving those puzzles is going to keep us busy for many years to come. But the basic phenomenon of growth and division is not so complicated. Given that, you can then ask, well, how could primitive cells start to control their growth, right? This is just happening based on the environment. For a living being to be adapted to its environment, it should be able to control its own growth. The first hint we had in that direction came from work by Irene Chen when she was in the lab. She showed that if you have a lot of RNA inside one of these vesicles, it creates a pressure on the inside that can actually drive growth. And it drives it in a very interesting way, in a competitive manner, so that vesicles with more RNA can grow by essentially eating vesicles that have less RNA on the inside. There's an even more interesting way that can happen that's shown in this diagram, and this came from thinking about a puzzle. If we begin with these very primitive membranes, they have properties that are ideal for a primordial cell, especially because nutrients can get from the outside to the inside and waste products can go out. They're very permeable to molecules. On the other hand, in modern biology, our membranes are made of more complicated molecules. They're very good barriers. Nothing gets across except if it's allowed to by complicated proteins that are pores and channels and pumps. So how could you transition from the primitive state to the modern state? So the logical inference would be some intermediate state where the membranes were mostly fatty acids, but maybe the cell had learned to synthesize a little bit of, say, two-chain phospholipids. That seems logical enough, but why would that happen? And why would cells that had these mixed membranes have any kind of advantage? Right? This progression could only happen if there's a selective advantage at each step. So Itai Buden, when he was a student in the lab, and he's now also a professor at UCSD, he did the experiment. He made vesicles that have a little bit of phospholipid. They don't do anything, but if you mix them with vesicles that are just fatty acids, the ones that have phospholipids start to grow, and the ones that don't start to shrink. So it's growth by competition. And the ones that are growing are doing it by absorbing molecules from the vesicles that don't have phospholipids. And in the bottom, you can see how a spherical vesicle has grown into a long filament. These are fragile, they're easy to divide. Okay, so from this kind of experiment, we've realized that the ability of a primitive cell for its genome to encode a ribozyme that could make phospholipids would confer a very strong selective advantage. But that would lead to an evolutionary arms race, because you always need to make more phospholipids than your neighbors in order to grow by stealing food from them. The problem is, or the interesting point is, that will drive cells to make more and more phospholipids, but then the properties of the membrane start to change. It becomes harder for molecules to get across. That means, on the one hand, it's now advantageous to have internal metabolism to make molecules that you can use inside, because they won't just leak out. On the other hand, it's also very hard to bring molecules in, and so we think that could actually be a driving force for the evolution of protein synthesis and membrane transporting proteins. Okay, So now I want to switch gears and talk to the genome, the RNA genome of primitive cells. And this is a really complicated topic. People have been working on it for over 70 years, but there's been a lot of progress from several different labs in the last decade. So I'll try to summarize briefly a few of the key advances that have been made. So first of all, what was primordial RNA look like? What did it look like? Is it the same as modern RNA? Or was it more heterogeneous? Did it have some different building blocks? There are reasons for and against all of these things. This is a very active area of research that we have focused on for the last many years. And so we're still trying to sort that out. But we can still think about the basic copying chemistry. And here, this work really began with the contributions of the late Leslie Orgel, who's really the father of this field of early RNA replication. And what Leslie realized was that the molecules that are used in biology, the triphosphates you see at the top, are great if you have very sophisticated enzymes that can catalyze reactions, speed them up. But they would be useless at the beginnings of life because we didn't have enzymes like that. They weren't evolved yet. So Leslie realized we needed more active molecules, things that can react spontaneously without enzymes, like the molecule you see at the bottom. Okay, so I just want to show you a video that shows you how we've been kind of thinking about this RNA copying. We imagine an RNA molecule floating around inside a cell in a very chemically rich environment filled with these activated nucleotides. And these activated nucleotides can find their partners by Watson-Crick-Franklin base pairing. So A with U, G with C. Then they click into place and build up, step by step, a complementary strand. And when that process is completed, you would have to pull the strands apart somehow, and then copy the copies and have a cycle of replication. Okay. So one of the things that held us back in making progress was the fact that copying at this stage doesn't work the way it works in biology. This is the way we thought it would work. It should work for many years. An activated monomer comes in, binds to the template next to the growing strand. One nucleotide gets added, and the strand grows step by copying the template strand. It turns out that it's much more complicated, much more interesting. So what actually happens is shown here. And if you like the detailed chemistry, you can see the molecules on the right. And if you like the diagram, that's on the left. So what happens is these activated nucleotides actually react with each other first, and they make this strange looking compound where there are two nucleotides that combine to the template. And the reason this makes the copying better is that now the substrate can bind by two base pairs, so it binds more tightly, and that means you need much less material, which is good. And also, this structure, this phosphate imidazole phosphate structure, in 3 dimensions is perfectly arranged so that the hydroxyl at the end of the primer can attack and drive the growth of the primer strand step by step, copying a template. So understanding this fundamental chemistry has been a big advance, and it's let us explore all kinds of questions about template copying, what kinds of alphabets work better, the accuracy of the process, and so on. So all these things we're continuing to work on. Copying a strand, though, and making a duplex product is not enough, right? As I said, you'd have to get those strands apart and then copy the copies and have a cycle. And that has been a very difficult problem to think about. In the diagram at the bottom, we just show that as you make a long double-stranded RNA molecule, it actually gets quite difficult to separate the two strands. And furthermore, once the conditions return to allowing base pairing, which you need to copy them, these complementary strands come back together very rapidly, thousands of times faster than our copying chemistry. So actually having a cycle of replication has been a problem that we haven't really known how to think about for at least 50 years. Okay. There are some other problems, like if you're copying a linear molecule, how do you start at the beginning and not start in the middle? So there's a whole bunch of problems like this that made the idea of doing replication just with physics and chemistry seem very difficult. Of course, if you have all kinds of elaborate, highly evolved biochemistry, it's easy. It happens in all of our cells. But how to do it at the beginning was a big problem. Okay. So we came up with a model, an idea of how to solve this problem during the pandemic. It was a time when everything was shut down. We couldn't go into the lab. We had to stop doing experiments. We were stuck at home. All we could do is think about different problems. We could talk to each other by Zoom. And so we came up with a model for how replication might work in very primitive cells. And it's a model we call the virtual circular genome model. And let me try to explain it. It's actually quite simple. The genome of a primitive cell would be represented by the green circle you see here. Okay, so it's a certain a list of the letters of RNA of A, U, C, and G in a circle. But in our model, there are no molecules that are complete circles. The only thing that needs to be present inside a primitive cell are fragments, short little fragments of RNA whose sequence maps to that circle. And the advantage of that is that all of these fragments can do multiple things. They can act as primers that get extended by our copying chemistry. They can act as templates that are what actually gets copied. They can play other functions that facilitate the process. And because the genome is a circle, there's no problem about where to start, right? You don't have to do it like in biology, where you start at the beginning and you copy all the way around the circle until you get to the end. Here, we do a little bit of copying all around the circle, also everywhere, all at once. Okay, so to show how that works, you just have this diagram to explain that. When these fragments come together, right? There can be hundreds of them. They can come together in many different ways by base pairing. Sometimes they come together in ways that allow a little template copying to happen. Sometimes they don't. But as long as you have a cycle where, say, some temperature fluctuations, high temperature, low temperature, these complexes can do a little copying, then they come apart. Then the temperature cools down, they come back together, you can do a little more copying. And if you keep doing that, you should actually be able to replicate the entire genome. So that is exactly the kind of thing that we're trying to demonstrate in the lab now. We have simplified models that verify all the component aspects of this. We've collaborated with other labs on simple models that show that the basic idea should work. And I just want to show one last experiment that, to me, is very satisfying. Because as I explained, we proposed this model to solve certain specific questions, right? How to actually do replication, how to avoid problems with knowing where to start. And the model solves that problem. But then it's turned out to have a completely different aspect, which is that it helps with the accuracy of replication in a way that was completely surprising to us. And this came out of work in collaboration with Arvind Murugan's lab in physics at the University of Chicago. And the idea is quite simple. If you consider a position in the genome in the wild-type normal sequence, that position can be copied. It's represented in many different fragments, and it can be copied by many different fragments that act as primers. But when a primer is being extended and an error is made, the wrong base has been put in, there are no primers that can copy that mistake. It can only get copied into the other strand after it's been extended, and that takes some time. And for many mutations, it can actually hardly ever happen. So this model that we've proposed also seems to help in an unexpected way, with the accuracy of transmitting information at the dawn of life. And that accuracy of transmitting information is an essential ingredient in the beginnings of Darwinian evolution that will lead to the more complicated forms of life that you heard Elena talk about. So just to summarize, we've obviously learned a lot of facts. We know new things about how life might have gotten started. But to me, one of the most general lessons is that to really make progress, we have to recognize when we're thinking about things the wrong way, we have to let go of preconceptions. And then that frees us up to think about things in different ways. I didn't do this work. It was all fantastic students, brilliant students and postdocs who have gone on to do their own amazing things in biology and medicine in many cases. And finally, let me just advertise my book that I wrote with my colleague Mary Olivio, who's an astronomer. He wrote about life out there in the universe, and I wrote about what I talked about here, how life might have gotten started when our planet was young. So thanks again for listening. It's been delightful to be here and talk to you all.